Please welcome MIT President Rafael Reif.
Boy, I can see that you need brunch this morning. I barely got a round of applause. Do it again, please. Come on. Yeah, thank you. Good morning, everyone. A pleasure to see you here again. I hope you enjoyed yesterday. I had a conversation with a couple of you last night, and a couple of you told me that, boy, yesterday was full of substance. And I hope that was a compliment, because MIT is all about substance, and this morning will not be the exception. By the way, talking about substance, one of the perks of my job is that there is so much exciting stuff going on at MIT, as you know. There is so much good, exciting stuff that's changing the world, that's shaping the future, that if you're curious, and I am, you want to know about new things, and you want to understand about what we're doing. And the very good news, the best perk of my job is that I can go to faculty and ask them what's going on here with this, and they have to be polite to me and explain that to me. And my concern is I gotta finish up my education by December 31st, because after that, they won't pay attention to me at all. So look, there is more substance for you this morning. We have a very nice conversation on the workings of the human brain. And you heard a little bit yesterday about AI and cancer. I mean, society, science has done a great job at keeping our bodies healthy for longer. And we all appreciate that, particularly those who are beginning their decades of the 70s like myself. So that is a great advance that we all appreciate. But as that's moving so fast and so well, reasonably fast and well, the workings of the human brain are still a huge frontier that is unexplored or ununderstood. And we're doing great work on that at MIT, and you're going to hear a great presentation on where we are on that. Another area that is extremely important is poverty, not just for all the right reasons of fairness in human opportunities, but for society and stability. I mean, poverty is a great indicator of an unstable society, and we have to address that. You're going to hear tremendous work MIT is doing in that area. And then, just for more fun, the first two are not that they are not fun, but they are serious stuff. And the third one is also serious, but a little bit more fun, at least to me, is exploration of the space. And I don't mean space like this one, I mean space like beyond the solar system, and how to do discovery in that domain. You're going to hear a great presentation from that. All these three faculty are award-winning faculty who, let me just tell you this much, they are some of the most faculty in demand we have at MIT, and yet they show so much respect and acknowledgement and gratefulness for all you do for us, that they are here on a Sunday morning chatting with you. So with that, let's get ready for, now I know that you need food before substance, so let's enjoy brunch and then we'll get the substance. Thank you so much for joining us. Thank you.
Please welcome Vice President Julie Lucas and Chancellor Melissa Nobles.
Good morning. My name is Julie Lucas, and I am the Vice President of MIT Resource Development. I hope that you all enjoyed the weekend here at MIT and had a chance to celebrate and learn more about how your support fuels our initiatives. This morning, we have gathered some exceptional speakers of our community to talk with you before you head home. I'm fortunate to have been a member of the MIT community since 2014. And since I arrived, I felt that there was something unique about MIT. I recognized very quickly that MIT not only draws diverse talent from around the globe, but also has a dedicated following. We're very fortunate to have such an engaged community of alumni, students, parents, staff, and institute leaders who are determined to make a better world. There's a spirit of general responsibility and care that is really wonderful, and it's something I see in all of our loyal and generous alumni and friends. All of you understand the importance of the core priorities of the MIT Campaign for a Better World: scholarships and graduate fellowships, faculty support, and investing in new spaces for learning and living. And the Institute is so very grateful for your continued commitment to what is at the heart of MIT's unique brand of teaching and learning. I am energized by the exciting prospects of all that lies ahead, not only for MIT, but for a world that benefits from the innovation that takes place here. Thank you for all that you did to ensure our goals were met and exceeded. While there is so much to be proud of, there is always more work to do, and we cannot build a better world without all of you.
I couldn't agree more. I am Melissa Nobles. I'm a professor of political science, and I have served as chancellor of MIT since last August. Before that, I was the Keen and Sahin Dean of the School of Humanities, Arts, and Social Sciences, and head of the Department of Political Science. In all of these roles, I've had the privilege of seeing the MIT community members recognize that science and tech need to be informed by the complexities of the human experience. The genius of MIT is to combine advanced science and technology with world-class humanities, arts, and social science research and education. All of it is focused on the complex cultural, social, and economic realities of the human world and the larger context of human society. Our responsibility is to prepare our students to face and to solve the world's biggest challenges. That responsibility is more important than ever. To do that, we must educate the whole student. We must inform and inspire students through multidisciplinary collaborations as the interactions between scientific, technological, and humanistic endeavors become ever more intertwined. Our students are deeply interested in STEM subjects, but they also recognize that there is so much more to learn about themselves, the world, and others. And the support that our students and faculty members receive from you make all of this indispensable learning happen every day at MIT, both inside and outside of our classrooms and labs. Like our talented students, the exceptional faculty members and researchers we will hear from today are facing the world's challenges head-on. Our first speaker is Abhijit Banerjee, the Ford Foundation International Professor of Economics. Professor Banerjee is the co-recipient of the 2019 Nobel Prize for his groundbreaking work in development economics research. In 2003, he co-founded the Abdul, Latif, Jameel Poverty Action Lab at MIT, otherwise known as JPAL, and he remains one of the lab's directors. Throughout his MIT tenure, he has been showing students showing us that economics is a field that can help us address some of society's biggest problems.
The power of traditional economics comes from, a lot of it comes from the fact that it starts from very specific premises about how human beings behave. Once you have those premises, then that often gives you very precise answers to questions about policy, which would be great, except that those premises are often false, and especially in the context of the lives of very poor people. And that means that this has consequences that can be catastrophic for the lives of many of the world's very poor people. The promise and the premise of the large-scale field experiments that we do in testing out policies is that it then allows us to look under the hood of policy without making those sometimes false assumptions about behavior. And this has allowed us to ask questions like, is this really how people behave? And if they don't, what do we do about policy? And this is the work we do in the Abdul, Latif, Jameel Poverty Action Lab. So let me give you an example. I mean, a very well-established proposition in economics even 20 years ago or 15 years ago was this idea that you should not make it too easy for people to do things that are good for them. And the reasoning is, if you do it, you will get people will take it. If you make it too cheap, they'll take it, but they won't actually take it seriously. They won't really use it, and so it'll be wasted. And that's sort of why things should be priced. They should be anti-malarial bed nets should have a price. Should not give people rewards for getting vaccinated. So all of that kind of very strong, clear prediction sort of guided the world of policy. Now, starting about 15 years ago, a set of large-scale field experiments started to append all that. And we started to learn, or maybe even 20 years ago, and we started to learn that, in fact, the people who are most subject to, whenever the price is a little bit higher, the kinds of people who actually don't want it tend to be the people who actually need it the most. These are the people who are often the world's poorest. They're often the most skeptical about, modern science, and therefore when you tell them that you have to pay $5 for it, they just walk away. And when you reverse that, you start to see that, they actually do take what's given. So if anti-malarial bed nets are given away, they actually take them and they use them, and lives get saved. If you, this graphic is telling these results from works of some of the JPAL researchers came in around the late 2000s. And you can see basically, and it sort of started to filter into policy, and you see that with that, you see an extraordinary decline in deaths from malaria. And among the groups that you would think would be saved by exactly that, people who are, children under 5 and children between 5 and 14, these are the people who sleep under those bed nets. And you see that suddenly the deaths have collapsed from something like 800,000 to closer to 400,000, or closer to 600,000. So that's the kind of thing that once you get the premise right, you can fix. Another example originally comes from Bangladesh, and it's the idea, the proposition that economists often started from is that you shouldn't make life too easy for poor people, because then they become lazy, and if they become lazy, then they stop doing the things that would make them richer, and therefore you always need to keep the pressure on. And that premise was a Bangladeshi NGO called BRAC kind of started what they call a graduation program, a program which took the opposite view, which is help people with a quick and easy start, give them an asset, give them some hand-holding to use the asset, and see what happens in a few years. Does actually let them take charge of their lives. And the remarkable evidence from that work that now we've studied in many countries is that, in fact, 10 years later, the people who got that one piece of help are 25% richer than the people who didn't. So it really has changed the way economists have thought about the world of the poor. And in particular, I think that has led to number of countries and now millions, or many millions of people being covered by these kinds of programs, where the starting point is actually to help people, not to tell them you have to try harder. And when we actually dig a little bit more into why we see that, why it happens that we manage to have these effects, it turns out that the critical piece is that poor people are often under enormous stress. They're always, if you ask them what they're thinking about the daily expenses. And you relieve that a little bit, and that opens up a mental space which makes them more productive. Now, once you go down that path, once it becomes, once you start thinking about the mental states of people, the fact that stress is unpleasant and maybe unproductive, and that anxiety is costly and depression is awful, I think you start to question this entire edifice that we've built in economics of valuing things by their market price. This is sort of the underlying premise of GDP, but also poverty measurement. The poor are people who don't have enough money, not people who are in generally disadvantaged by life and under stress and unable to function. And that, I think that general premise of valuing things by what people are willing to pay for it, of course, is immediately undermined when you think about, some of these anti-malarial bed nets, which will save lives, but people are not willing to pay for. Or in reverse, when people buy things, I think the premise is often that this is something that if they're buying it, it must be valuable to them, but in fact often they buy it because others are buying them. And I think both sides of that proposition, so sort of this equation of value to what the price people are paying, has led to questioning of that has led to this enormous effulgence of research on what is called behavioral economics, so thinking about where does welfare come from, what makes people happy. And I think I will now invite my colleague Frank Schilbach, who's one of the young leaders in that field, to tell you a little bit about his work and the excitement that has brought into the field of economics. And so I think here is Frank.
The measurement of poverty has focused largely on material conditions, based on the idea that people need enough money to eat, and if you don't have enough money to eat, you must be poor. But being poor means so much more than not having enough money to eat. In many cases, it means facing challenging sleeping conditions. It means sleeping on the floor with many people in the same room, facing dust, noise, mosquitoes, or heat, or attention or worries, all interfering with people's good night's sleep. The poor are also disproportionately affected by physical or psychological pain, often feeling lonely or excluded. They often, being poor means struggling with your everyday finances, worrying about paying your kids' school fees, while at the same time even struggling with depression or anxiety caused by these circumstances. Each of these factors might be important contributors or barriers to people taking advantage of high return investments such as bed nets. They might also inhibit people from working productively at work. And we should care about that, and that's very important. However, each of these factors are also inherently important in and of themselves. We should inherently care about whether a person is able to enjoy a good night's sleep, whether they experience depression or anxiety, or whether they're suffering from physical pain. These factors are key predictors of people's well-being, and they're all disproportionately affecting the poor. Now, until recently, measurement has been a key obstacle in this space. However, recent advances in technologies have greatly improved our ability to measure these types of factors, and we have been able to do this with sometimes striking insights. For example, objectively measuring people's sleep in their homes using wristwatch-like devices, we recently discovered that the urban poor in India sleep about 5.5 hours on average, and the sleep is highly inefficient and of low quality, disrupted by over 30 awakenings in a typical night. Let me show you what a typical night looks like for a typical person, low-income person in Chennai, India. This person went to bed at about midnight, spent about 8 hours in bed, so spent about 8 hours in bed and got up at about 8. Within those 8 hours, the person slept about 5 hours, which you can see here in the gray bars. This is time that the person was asleep. But you can see that this night is riddled with interruptions, with awakenings, which you see here displayed in red. Now I want you to think about for a minute, how would you feel, how exhausted would you be after one such night that you experience in a given night? And then think about if every night of your life looked like this. And remember, this is a typical night for a typical person, typical low-income person in Chennai. Now, we can go further and measure other aspects of poverty too. For example, biomarkers of stress. Recent work has shown that cortisol levels indicating stress are higher after negative income shocks among farmers in Kenya. And providing cash transfers increases happiness and lowers such stress as measured by reduced cortisol levels. There are also big improvements in detecting depression and anxiety via self-administered and locally validated questionnaires in low-resource contexts. These surveys are highly predictive of diagnosis of depression and anxiety by trained psychiatrists, and they show that the poor, and in particular women, are disproportionately affected by such conditions. But we can go further than just measuring these factors. Simplified, inexpensive, and scalable therapy is very successful at improving mental health in low-resource contexts, as recently shown in India and in many other settings. Now, a key goal now of future poverty research is the systematic and high-quality measurement of well-being and its determinants in low-income contexts, such as sleep, mental health, physical pain, or loneliness. We may or may not want to go all the way towards measuring and using gross national happiness instead of GDP, as in Bhutan, but we surely want to further improve the measurement of these factors and of well-being more broadly. This improved measurement, when they'll help us understand and even perhaps redefine what it means to be poor or what our concept of poverty is, will dramatically improve our understanding of the psychological lives of the poor and how we might be able to improve their lives and their well-being. Crucially, interdisciplinary collaborations between psychologists, economists, and neuroscience are the key way forward towards achieving this goal. Thank you.
Our next speaker is Anne Graybiel, Institute Professor in the School of Science, an investigator at the MIT McGovern Institute for Brain Research, and the recipient of the 2012 Kavli in Neuroscience. Her lab focuses on the brain structures that are linked to a number of diseases and disorders, including Parkinson's disease, anxiety, depression, and addiction.
Thank you very much. In this little time, I want to talk about an issue that's deeply embedded in our beings, and that is the ability for us to decide to do something and then actually to do it. To me, this is like a miracle, and all I can say is that now is a miraculous time in neuroscience when we may be able to get at some depth into what controls our decisions and how we can improve them. We know that motivation has a lot to do with this. It's crucial. And we also know that through this kind of looping which we have in our brains, we can somehow produce actions, movements, but also thoughts. And I'm showing you here an image, a very famous image of the brain. It's a PET image, and the peak shows you where the dopamine transmitter is, the transmitter that has so much to do with the control, the modulation of our thoughts, our emotions, and our movements. Now, you heard a little bit about reward already. I want to show you that here is a scan, another, this time an activity scan of a human brain. And it has to do with how our brains respond to reward. And for those of you who are deeply embedded in mathematics, here's that equation that Professor Banerjee spoke of. And so I want to show you the remarkable effects of this thanks to our brains. This is the first time this little girl has ever pressed that pedal. And you can see she goes right back to it. So we can learn from these reinforcing experiences, and that's a wonderful thing. But there is the other side of this whole issue, and that is that if these loops are in some way broken, then we can face neurodegenerative diseases. Here are extrapyramidal disorder diseases like Parkinson's, Huntington's. but also a number of neuropsychiatric disorders now turn out to be related to those deep brain regions that I showed you. And also, just the way we could learn, you just saw an example of it, the kind of excessive habits that we can pick up. And then, as you just heard about, the effects of stress, which can be terrible. So I'd like to give you just a tiny glimpse of how this deep brain structure, now I'm showing you a mouse brain, the nose is up, the back of the brain is down, and we've engineered in a marker of red, and you'll see these red cells which are in the same kind of pink deep zone I showed you before in the human. And you'll see that the red cells make up a labyrinth of zones within this deep brain structure, a remarkable labyrinth that picks out certain spots. And now I want to show you that labyrinth again, but now the cells are in, oops, sorry. Now the cells are shown in white, and as this goes around, I want to show you how their outflow goes. It goes deep into the midbrain to these strange looking places. So what I've shown you in those original red cells are regions that receive input from mood-related neocortex that could affect our affect, our emotions, our states. And now what I'm showing you is where they go, where they project to. And now I want to blow up one of those strange-looking places. And what this shows you is in an engineered mouse, the red cells are dopamine-containing neurons way down in the midbrain. And the stem shows you, the stem of this bouquet, shows you their receiving zones going down. And those labyrinths I showed you send their long fibers down to this region, and they come up the stem, and they encircle and entwine themselves with the dopamine-containing receiving zones and make contacts, and now we know they can actually control the activity of the dopamine-containing neurons. So there is great potential in trying to understand this great circuitry because it's so important as a modulator. Our brains have hardwires, and then they have amazing modulatory systems that are just now beginning to be studied. And it's a shame and a terrible thing because these very circuits are hit in neuropsychiatric disorders. They're studied with receptors for drugs such as LSD. And we have every reason to hope that MIT can be a true leader. There's enormous potential in learning how to bring knowledge of the brain to help people throughout the world. And I want to bring on just the sort of person who can really build this future, Emily Hoekstra.
Thank you, Anne. After 4 years of working with Anne, we just discovered in preparing for this talk that we got started in neuroscience for the same reason. Both of us came from physics and chemistry, but we eventually became fascinated with understanding free will and volition. Speaking of free will, you may recognize a jar of marshmallows on the screen. It's a reference to the famous marshmallow task, in which children were offered 2 marshmallows if they could delay their gratification for the first one, or they could eat one immediately. This task was aimed to capture in the lab the kind of cost-benefit trade-off decision-making that is common in the life of every human. I generate laboratory tasks like this one, but for mice, so that I can engage specific brain circuits and watch what the neurons in those brain circuits are doing during those tasks. Sometimes I turn on or off certain neurons to test whether they're involved in generating a given behavior or changing it. We use decision-making tasks because in mice, as in humans, cost-tolerant or risk-averse behavior in an individual reflects a lot of cognition as well as mood function. The decision thresholds can be altered by turning on or off the set of brain circuits that we study. And these circuits and these behaviors are dysfunctioning in diseases such as Huntington's disease and Parkinson's disease and the others that Anne mentioned. We have done the deep neuroscience to understand that these circuits span from the prefrontal cortex, smart stuff up at the front, which sends connections down to make its earliest connections in striatum on the islands that Anne showed you. We know that happens about mid-gestation, and that those labyrinthine islands are unique in their ability to send their own connections down to drive dopamine neurons, a neurotransmitter that is involved in reward seeking, risk taking, and motivation. Dopamine is the neuromodulator that I would argue holds the most sway over those behaviors that guide the success of the individual and the survival of the species. In approaching diseases and disorders of the brain, what excites me as a systems neuroscientist is that, especially now at this time, we're sitting at a golden time, at the culmination of incredible parallel developments, sometimes after decades of deep research in the field of neuroscience, which has given us the ability to turn on and off neurons at will and see what those neurons are doing in great detail. In the field of genomics, which has broken open the mysteries of what genetic mutations are associated with a given brain disease or disorder, and in gene therapy. And this parallel deep research is now starting to pay dividends. As an example, we have done the science to know that in Huntington's disease, terribly debilitating and eventually fatal disease of motor degeneration that is genetically inherited and strikes after childbearing years, we have discovered that in Huntington's disease model mice, the signals coming from those islands between the smart stuff and dopamine are trounced by this disease, and their decision boundaries show they are deeply cost averse. But we are testing a solution. Across fields, we forge collaboration and iteration between those like us who understand the systems and brain circuits and the timing and mechanisms of the disease process, and those who are developing safe delivery methods for therapy to the brain, shuttles that can be injected in a vein and can miraculously cross the blood-brain barrier, a development of only the last few years. And lastly, those that are specifically designing the payload that gets delivered, the actual gene therapy, shown in this brain section in magenta. You can see that it covers the entire span of the mouse brain. We are experts in using mouse disease models. We have designed tasks that get at the problem of a disease, and now we are able to deliver systemically a gene therapy treatment intravenously to specifically target the one bad copy of the gene, leaving the other one alone. And incredibly, we are finding that the responses of our striatal island neurons are recovering, and that the decision boundary is moving from being highly cost-averse back in the direction of healthy. I'm excited because HD research. I'm excited, hear me? Because HD research is an example of what's to come. Unlike the other diseases and disorders we study, it's caused by a single gene. It is no less terrible for its genetic simplicity. but it is among the most straightforward for which to develop and refine a solution. There is still much to be done in developing effective therapies for HD. But 2 years ago, for the first time, HD families saw the first therapies progress into clinical trials. Now it is my hope and expectation that HD research is only the beginning and that it will lead the way forward. As geneticists understand more about mutations that occur in, for example, autism spectrum disorder, we systems neuroscientists are working fast to understand where in the brain, how in individual neurons, and often when in development, those mutations are wreaking havoc. In our lab, we're doing the same kind of deep research in models of Parkinson's disease. Autism, which, according to the CDC in 2021, is affecting now 1 in 44 children, up from 1 in 88 just 5 years ago. We're working with models of obsessive compulsive disorder, models of addiction, as well as depression and anxiety, all with the goal of understanding the circuits for developing and delivering effective treatments. And so, in an iterative manner across these adjacent fields, we are and will be making faster and better progress to deliver gene therapies to the right brain system, to the right type of neurons, at the right time to garner improvement in a disease or disorder. MIT is a stunning place of innovation and collaboration, and I truly believe there's no better place to work on these solutions. Thank you.
Our next speaker is Sarah Seeger, the class of 1941 professor of planetary sciences and a 2013 MacArthur Fellow for her work in physics. She has pioneered many research areas of characterizing exoplanets with concepts and methods that now form the foundation of the field of exoplanet atmospheres. Her present research focus is on the search for the wave of exoplanet atmosphere. Look, I'm a political scientist. Our present research focus is on the search by way of exoplanet atmospheric biosignature gases.
Hello. So, 2 years ago, I did something that shook the world of science, planetary science. It involved taking risks and a fresh perspective that led to innovation in true MIT spirit. So let me take you on a journey of exploration. So it starts with a backstory from a very long time ago, billions of years ago, when a simple single-celled bacteria, cyanobacteria, figured out something very clever, how to harness energy from the sun, photosynthesis. And in the process, generated a waste gas, oxygen. We all need oxygen to breathe. And eventually, that oxygen filled our atmosphere to 20% by volume. But oxygen is so highly reactive, it shouldn't be in our atmosphere at all. Without plants and photosynthetic bacteria, we would have no oxygen. That's the heart of how we hope to find signs of life beyond Earth. By looking at planets far away, planets around other stars, we call them exoplanets, and to look for gases that might be attributed to life. So what I love to think about when I look at the night sky is that if there's an intelligent civilization on a planet orbiting a nearby star with the kind of telescopes we're hoping to build looking back at us, they'll be suspicious there's life here. Not by city lights or the Great Wall of China, but by oxygen, a gas that doesn't belong, that's not made by large amounts by volcanoes or lightning, but only by life. So the controversial thing I did, I was a part of a team that made a big announcement about 2 years ago about a small amounts of gas on another planet, a gas that doesn't belong, that as far as we can tell is not made by lightning or any chemistry we know about. Maybe some of you heard about this. The gas is called phosphine, and the planet is Venus, our sister planet. Now, why is this so controversial, so crazy and out there? Because you probably already know that Venus is a terrible place for life. It has a massive carbon dioxide greenhouse atmosphere, and that surface is too hot for life of any kind. So like the molecules we need for life, they're just not going to be able to be stable at those temperatures. But just like here on Earth, if you hike up a mountain or go high up, it gets colder. And so too on Venus. So far above the surface, in the cloud layer, about 50 kilometers up, it's about actually the right temperature for life. And this idea, the concept is that there might be, it's like a super crazy idea still, okay, but that there might be like microbes floating around in the clouds. And this idea has been around for over half a century since Carl Sagan first proposed this. And here on Earth, by the way, we also have microbes in our clouds. They get swept up by winds, they stay aloft for about a week or so, and then they get dropped back down. So Venus is still a terrible place for life. It doesn't matter. These clouds are not water. They're sulfuric acid. And they are, there's a lot going on. I wish I could tell you, I wish I had more time, because there's a lot of new developments. Because of phosphine, we have motivated people to make like fat membranes that can survive in sulfuric acid. We have found rich chemistry in sulfuric acid. But I only have 6 minutes, so. So what I did, but about life or signs of life, no matter how crazy it is, the amazing thing is, Venus is right next door. We can, the best way to pursue this idea that there might be signs of life or life on Venus is to go to Venus and to send an atmosphere probe to go directly down in the atmosphere. So during the pandemic, I actually, I assembled a team. Because like this topic, it's still too taboo for like big government space agencies like NASA. So I assembled a new team to make this happen. And one, maybe slightly controversial thing I did with my team was I picked like the top, very best planetary scientists and engineers. But with one exception, I purposely avoided anyone who worked on Venus. I also excluded the cranky older people. Yeah, I'm one of those in my like original field of exoplanets, and because we didn't need entrenched views or people forcing their views, we had to like think with a new perspective and as freshly as possible. So we had to do a lot of hard work since we had to essentially like relearn everything from scratch. But I'm very proud of my team. We did some amazing things. We invented new instruments. We looked at old data from the 1970s and 80s when the Americans and Russians sent many probes down through the atmosphere. We found data that has been discarded, like ignored, such as tiny amounts of oxygen, tentative detection of ammonia, and a depletion of sulfur dioxide and water vapor in the cloud layers. And it's really interesting because we also found that current models of the atmosphere, they don't work, but as long as people state what they're doing to make the models work, it's okay. But these models are still commonly used. So we made a new model to explain the old data. And just like oxygen on Earth, the best explanation might just be life on Venus. I mean, sometimes I don't know if I'm on a wild goose chase with this project or if it's really the best thing ever. And this is really the spirit of MIT, and that what all of you bring and continue to support, to think outside the box, to take risks, to make new things happen. So back to my specific story. We're going to Venus. One of the outcomes of my 2-year study is we're going to Venus with a new type of focused mission. We're going with Rocket Lab, partnering with Rocket Lab, a company in the new space. It's the first private space mission, and we have a target launch date of May 2023. This is like a small mission on the cheap as far as space missions go. So it's going to go to Venus, drop a probe, which will go down through the atmosphere, like without a parachute. It'll take about an hour, but it only will spend 3 minutes in this cloud layer. And we have chosen one specific science instrument that will shine a light through a window, and it'll measure the backscattered polarized radiation, but in addition, it's looking for fluorescence, which could indicate the presence of organic molecules in these cloud particles. Doesn't mean there's life there, but it's enough to like motivate the search to keep going. We've inspired more traditional NASA and ESA missions to be selected. They can't look for life, it's too controversial. We have future mission plans that we're still trying to develop. So in closing, I invite you to reach out to me by email to somehow join my exploration of Venus. It's a vast universe out there just waiting to be explored. So I wish you clear skies, clear thoughts, and your own continued journey of exploration. Thank you very much.
Thank you to all our speakers for sharing your inspiring work with us. There's so much work to be done, but there are so many reasons to be hopeful and excited about what the future holds. I want to thank all of you, our distinguished faculty speakers, and our loyal supporters for providing a foundation for MIT students to form and pursue new ideas and then take those ideas out in the world and transform it for the better.
Yes, thank you everyone for attending the MIT Better World Weekend. This weekend has highlighted the exciting education, research, and innovation happening at the Institute. And today's program was no exception. The campaign for a better world may have concluded, but as you've heard here today, the pursuit of a better world continues. Your generosity contributes directly to making MIT the powerhouse it is for problem solving, for educating wise citizens, and for making a better world. We are enduringly grateful. So I invite all of you to continue discussing these ideas with the MIT community members at your tables. Thank you and enjoy.